Next, we need to address eicosinoid synthesis. Now, the purpose of eicosinoids are local mediators of information. So we've seen so far with insulin and glucagon, these are endocrine hormones that are able to communicate information in the body throughout the entire body, that being either a fed or fasting state. But occasionally we need to have local mediators, local information to provide for adjacent cells, and this is typically involved in inflammatory reactions. So inflammation. What this typically means is that when there's an inflammatory event, those inflammatory cells need to put out information that are important to the adjacent cells and altering physiology during the purpose of the inflammation. So we're going to start out with the fatty acid arachidonic acid. This structure here is the typical representation of arachidonic acid. It's 20 carbons long. It has 4 double bonds, so it is a polyunsaturated fatty acid. Double bonds are located at carbons 5, 8, 11, and 14. Now, this fatty acid is not synthesized by humans or mammals because we have double bonds positioned greater than carbon number 9. Remember, we can produce double bonds up to carbon 9, but not beyond. So this is derived from a plant source. Now, once this is taken in a dietary fashion, this fatty acid can be stored as a phospholipid. So remember, we have our head group on the phospholipid. We have the fatty acid coming off of the glycerol backbone, and one of these fatty acids can be represented by arachidonic acid. So arachidonic acid can be stored in the plasma membrane. Now, when we have an inflammation response, these inflammatory signals will introduce esterases, enzymes that will hydrolyze ester linkages. Typically these are lipases, and it will release the fatty acid from the membrane. The fatty acid now can be processed. And processing includes taking the arachidonic acid and turning it into either prostaglandin or thromboxane. The enzyme that does this is called cyclooxygenase. And this enzyme converts arachidonic acid into prostaglandins and thromboxanes. And the thromboxane is involved in platelet activity. And what this primarily does is promote platelet aggregation. Prostaglandins are involved in inflammation. So these prostaglandins are essentially the inflammatory signals, and so these are considered pro-inflammatory because they enhance the inflammatory signal. Now, arachidonic acid also has another processing pathway. We won't address this one, but just for completion, this one is producing another small molecule called leukotrienes. And leukotrienes are processed using arachidonic acid by the enzyme lipoxygenase. So it's a different enzyme taking the arachidonic into a different pathway. Again, it's going to stimulate leukocytes, so it's again part of the inflammation reaction. Now, the part that needs to be addressed at this point is this becomes a very interesting pharmaceutical target. In order to control inflammation, we can think about producing an inhibitor for the cyclooxygenase enzymes. This is important, for example, in rheumatoid arthritis. Rheumatoid arthritis is an inflammation of the joint. Many, if not all of us, know someone that has suffered or is suffering from rheumatoid arthritis, and this is a very painful, life-changing situation, so we need to find a way to control the inflammation that is causing the arthritic pain in these joints of these individuals. So technically, if we could develop an inhibitor of cyclooxygenase, we could prevent the formation of prostaglandins. These inhibitors are known. One is aspirin. The other is ibuprofen, naproxen. These are all part of the class of drugs called NSAIDs, non-steroidal anti-inflammatory drugs. These are very powerful agents in those that are having problems with rheumatoid arthritis. If they take any one of these agents inhibit cyclooxygenase, prevent the conversion into prostaglandin, and reduce inflammation in rheumatoid arthritis. Now, individuals after having a heart attack are recommended to take aspirin at low doses. Well, with a heart attack, heart attacks increase the chances of having a stroke. And a stroke is essentially a clot that's broken free. So the concept of a low-dose aspirin is to inhibit the cyclooxygenase, thereby reducing the concentration of thromboxane being produced, which will essentially decrease the amount of platelet aggregation and prevent the formation of a stroke.